I'm going to go ahead and record this. All right, so when I introduced the whole concept of visualization, and on that very first day of class, I was defining visualization. You may recall that, different people have different definitions of what visualization is all about, but I use a fairly narrow definition, which simply means, visualizing the invisible, illustrating things or representing things visually that can't be seen, that are somehow invisible to the naked eye. Now, that doesn't necessarily just mean small things. It could involve things that are so big that you can't take it all in, like a map, in a sense, is a visualization, because you can't, go up in an airplane or a satellite and view a satellite view of the Earth. So a map helps you visualize geography and where things are located on the Earth, or a topographic map that shows elevation and things like that. Those are examples of visualizing things that are unseen, because you just can't usually get that mapped view, that bird's eye view of the world. But it also includes things like looking inside the body, like we did with Horos, things that are large enough to be seen with the naked eye, but they're invisible because they're hidden inside the body. So we use X-rays and CTs and MRIs to look inside the body. But now I want to start getting into things that really are just too small to be seen with the naked eye. They're invisible simply because they're too small to be seen with the eye. So I want to review units of measurement. Hopefully this is all review to everybody, to talk about, just how small is small. How small are we talking about before things become invisible? What kind of scale are we dealing with when we talk about looking at cells, organelles, or even things like individual molecules? So here we see a list of the common metric units that are universally used in the sciences. And I feel like, if you don't already know these units, you should familiarize yourself with them, because you'll be exposed to this stuff throughout your career as medical illustrators. Medical illustration is about a lot more than just illustrating anatomy and surgery and things like that. You're often illustrating things at the cellular level or even the molecular level. So you need to be familiar with these units of measurement and these, the different levels of scale. So I think most of us have kind of an intuitive understanding of what a meter is. A meter is a little bit bigger than 3 feet, so it's a little bit bigger than a yard, which is another, English term or American term. So, the height of a small child, might be like 3 feet tall, something like that. A centimeter is 1/100th of a meter. So there are 100 centimeters in a meter. So in scientific notation, we would say that a centimeter is 10 to the minus 2 meters, or 1 over 10 to the 2, 1 over 100. So 1/100th of a meter. And there are 2.5 centimeters in an inch. So you could say that a centimeter is roughly half an inch, or a little bit less than half an inch. And I said width of a pencil here. That's actually a pretty fat pencil to be about a half an inch. But, something maybe between the width of a pencil and the width of your pinky finger or something like that would be a centimeter. A millimeter is 1/10th of a centimeter, or 1/1000th of a meter. So there are 10 millimeters in a centimeter, 1,000 millimeters in a meter. So we would say that a millimeter is 10 to the minus 3rd meters, or 1 over 1,000. And of course, the abbreviation would be mm. And, I gave some examples here. An ant, although this would actually be a very small ant. Ants come in a lot of sizes. There are ants that are like 2 inches long. They're great big ants in the Amazon and stuff. I'm not talking about those big ants. Even like the carpenter ants that you might see, hopefully not in your house, but you might have them. Some of those carpenter ants can be 1/2 inch or 1 inch long. So those would be big ants. I'm talking about the really tiny, tiny little ants. Or maybe a better example of something that's on the scale of a millimeter would be the head of a pin. even the head of a pin, maybe 2 to 3 millimeters in size, depending on how big the pin is, but something in that scale. A common unit of measurement that's used in the biological sciences is a micrometer, or frequently referred to as a micron, represented by the Greek symbol mu, or mu and then the m. A micrometer is one millionth of a meter, or 10 to the minus 6 meters. Also, you could say that it's 1/1000th of a millimeter. So it's very small. Now we're getting down to things that are microscopic in size, that you cannot see with the naked eye. Red blood cells, which are relatively small as cells go. A typical red blood cell is only about a micron, maybe a few microns in diameter. So we're getting down to the cellular level now. The next smaller unit of measurement that's commonly used in the sciences would be the nanometer, which is 10 to the minus 9th meters. So one billionth of a meter, or 1/1000th of a micron, and just represented by the abbreviation NM. And now we're down to the molecular scale. The DNA double helix is, approximately a nanometer, or I think it's about a nanometer in diameter, maybe a little bit less than a nanometer in diameter. But we're down to the scale of individual molecules, like the DNA double helix or something like that. And then finally, even smaller than that, 1/10 of a nanometer is called an angstrom. So 10 to the minus 10 meters. It's represented by this symbol with a capital A with a little circle over the top of it. And now we're down to the scale of individual atoms. An individual hydrogen atom is approximately an angstrom in diameter. The human eye is capable of seeing something that's about the size of 1/10 of a millimeter, 0.1 millimeters, or 10 to the minus 4 meters. So like 1/10,000th of a meter. Now that's approximately the thickness of a human hair. So, of course, you hold up a human hair in front of you, most people can see that, but you get anything significantly smaller than that, and it's just not going to be visible to the naked eye. So anything below about 10 to the minus 4 meters, you need some form of magnification to be able to see it. Now, in addition to this chart that just shows these common units of measurement and, how they relate to a meter, and, some objects, just for scale. The other thing I've included on this slide off to the right is an image showing the different wavelengths of different forms of electromagnetic radiation, abbreviated ER there. So there are many different forms of energy that we call electromagnetic radiation. And the only differences between these different forms of electromagnetic radiation is the wavelength. So electromagnetic radiation refers to a form of energy that travels in a wave form. And what distinguishes one type of electromagnetic radiation from another is simply the wavelength, which is literally the distance between the peaks of the waves. So you'll notice on this scale, up near the top, you have things like radio waves, which are actually very long. There's a couple other things I didn't put on here. Microwaves, like your microwave oven, have very long wavelengths. They would actually be above radio waves. In fact, microwaves, the wavelength, the distance between the crests of the waves can actually be measured in centimeters or even in meters. There's literally that distance between the crests of the waves in a microwave radiation. Even radio waves, there's what's called shortwave and longwave radio. You may have heard of something called a shortwave radio. It's just referring to the wavelength of the specific radio signal. Then you get down into forms of electromagnetic radiation that we refer to as light. There's infrared which people will refer to that as light, even though infrared light, in quotes, is not actually visible to the eye. It's so close to the wavelength of visible light that we will still sometimes refer to infrared as a form of light. Likewise, ultraviolet. Ultraviolet, we often refer to that as a type of light, even though it's not visible to the human eye. There are lots of other animals that can see ultraviolet wavelengths of light. Insects in particular are known for being able to see into the ultraviolet range. That's how bees locate their flowers for pollination. They can see markings on the flower that are visible in ultraviolet light that aren't visible to the human eye. But visible light, shown there by the little rainbow, is a form of electromagnetic radiation that falls in a very specific range of wavelengths. The crests of the waves in visible light range from about 400 nanometers. So 400 nanometers, if a nanometer is 10 to the minus 9, you multiply that by 100, you get 10 to the minus 7. So, 4 times 10 to the minus 7 meters for what would be violet light or purple light, all the way up to 700 nanometers for red light. So that would be, 7 times 10 to the minus 7. But what that means is that all visible light falls in the range of wavelengths somewhere between 400 and 700 nanometers. And if you have electromagnetic radiation that's either, has longer wavelengths or shorter wavelengths than that, then our eye can't detect it. So it's not visible to the eye. So the eye only detects wavelengths of light that fall within that narrow range of the electromagnetic radiation spectrum. There are other forms of electromagnetic radiation that have even smaller wavelengths. You might not think of an electron as being a form of energy. We often think of an electron as being a particle, but it turns out that all of these forms of electromagnetic radiation have what physicists call wave-particle duality. They have aspects of their behavior that function kind of like particles, and they have aspects of their behavior that function kind of like waves of energy. So even visible light, we talk about photons of visible light as if it's a type of particle, and yet visible light definitely has a wavelength. Same thing with electron. We think of it as a particle, but it also has a wavelength. So electrons have a smaller wavelength, even smaller than ultraviolet light. And then X-rays have an even smaller wavelength than that. And I plotted them on here approximately where they would fall relative to our units of measurement. So visible light, like I said, it's between 400 and 700 nanometers, so somewhere in the range of 10 to the minus 7 meters. So that means that the wavelength of visible light is smaller than a red blood cell, but bigger than a DNA molecule. The wavelength of electrons is actually a little bit smaller than the diameter of the DNA double helix, and X-rays are even smaller. X-rays have a wavelength down around 10 to the minus 11 meters, so like a 1/10 of an angstrom. Now why am I telling you all this? Why does this electromagnetic spectrum stuff matter? Why is it important? Because there's a general principle in physics that electromagnetic radiation can only reflect off of an object if the object is bigger than the wavelength of that form of electromagnetic radiation. So that means that visible light can only reflect off of things that are bigger than 10 to the minus 7 meters in size. So visible light can reflect obviously off of the head of a pin or an ant. Visible light can reflect off the surface of a cell like a red blood cell, because those things are bigger than 10 to the minus 7 meters. But when you get down to things smaller than that, like a molecule like DNA, visible light cannot reflect off of the surface of something that is smaller than the wavelength of that form of energy, which means that it is theoretically impossible to see a molecule because light can't reflect off of the surface of an individual molecule. And I think I've made that point in here before, that if light can't reflect off the surface of something, you can't see it, because there's no way for that light to reflect off of the object and reflect back to your eye for it to be visible. So light cannot reflect off of anything smaller than a small cell or a small organelle. So anything smaller than 10 to the minus 7 meters. So it is theoretically impossible to actually see anything down at the molecular scale or the atomic scale. So what that means is, with light microscopy, when you look at something under a microscope, it doesn't matter how powerful the microscope is, it has a theoretical limit of how small something is that it can see. A light microscope can only see things down to about 1/10 of a micron, down to about 10 to the minus 7 meters, because then you reach the wavelength of visible light and you literally cannot see anything smaller than that. So let's talk a little bit about light microscopy, because in order to see anything smaller than a human hair, you need to use some kind of magnification. And the oldest and still one of the most popular forms of magnification is light microscopy. The light microscope's been around for a long time. Easily goes back into the later part of the 1500s. You may have heard in a biology class, you may or may not remember this name, but there's somebody named Anton van Leeuwenhoek, who was famous for using a light microscope to look at little single-celled organisms in pond water. I think a lot of people assume that he was actually the inventor of the light microscope, but he wasn't. I believe van Leeuwenhoek was actually working in the 1600s, and light microscopes had been around for at least 50 or even 100 years by the time he was using them. We know from historical records that Galileo owned at least one light microscope, and used to, just for fun, would look at little tiny things under his light microscope. Light microscopes are amazing. I mean, they've contributed just, in ways you can't even imagine, contributed to our understanding of science. I mean, we wouldn't even know that there was such a thing as a cell if it wasn't for the light microscope. But despite all of the wonderful things about them, and I won't try to list all the great things about them, hopefully you have some general idea of how important the light microscope has been in the history of biology, but there are also some limitations to the light microscope. One of them I just discussed, the fact that the resolution of a light microscope, that the size of things that it can see, is limited by the wavelength of visible light. So light microscopes can only see things down to the scale of 10 to the minus 7 meters, or about 1/10 of a micron. So, smaller than a cell, but bigger than an individual molecule. So light microscopes are great for looking at whole tissues. If you want to look at the way cells are organized to form, an epithelial tissue or some other organ tissue, those of you who take histology and histopathology next year, that's what that whole class is all about, looking at microscopic images of tissues and organs to see, the cellular structure of tissues and organs. You can see organelles within the cells, although just barely. I mean, you can tell that individual cells have a nucleus. You can tell that they have a Golgi body and an endoplasmic reticulum and all that kind of stuff. But you can't get a really good view of the organelles. You can't make out their detailed structure. Bacteria are barely visible under a light microscope. You can usually see individual bacteria, but you can't necessarily make out much about the detailed structure of the bacterium. And things like viruses are usually completely invisible to light microscopes. Viruses are simply too small. They are, orders of magnitude smaller than a typical cell in the body, so they're not visible. And as I've already mentioned, individual molecules are also not visible under the light microscope. One of the other downsides to light microscopy is that normally when you want to look at something under a light microscope, you slice it very thin. There are exceptions to that. There are other kinds of light microscopes that let you look at things reflecting off the surface of an object, but most of the time when we use a light microscope, we have to slice our specimen super, super thin, put it on a slide, put a little cover slip over it, and then set it onto the microscope. In fact, if you look at the picture of the microscope here, you'll see that number 7 is a light source. So there's actually a light built into the microscope. That light shines up through the slide. The slide would be set on this little platter here. The light would shine through the slide, through the objective lens, and then up through the ocular lens up here at the top to be magnified. But what that means is that the slices that you're creating have to be so thin that they're almost not visible. If you just took a thin slice through, some tissue or organ or something, or a little slice through a piece of a plant or something, put it on a microscope slide and looked at it under the scope, you'd hardly see anything, because it has to be so thin to let the light through that it's virtually invisible unless you stain the sample. So to make things visible in light microscopy, almost always the sample has to be stained. And the stain then colorizes the sample so that you can actually see what you're looking at in these super thin slices. So I want to spend some time talking about these different techniques that are used to stain microscopic samples, because as medical illustrators, you will run into these things all the time. And it's really helpful to have some understanding of what the heck you're looking at when you look at a stained sample. One of the first things to recognize is that almost always when you look at what's called a micrograph, in other words, a photograph of a microscopic image, almost always you're looking at a stained image. So the colors you're seeing are not the actual colors of the organs or the tissues or the cells. What you're seeing are the color of the stains that have been used to dye those cells so that they become visible. And by far the most common stain that you will encounter as medical illustrators is what's called H&E stain technique, which is short for hematoxylin and eosin. So it's a mixture of two different chemicals that stain different structures in the cells. And they stain them with very characteristic colors that make things very easy to distinguish when you look at them under a light microscope. The H&E staining method is definitely the most common stain that's used in histology. So if you are looking at the microscopic structure of tissues and organs, usually they're going to be stained with H&E. Also in the field of pathology, looking at diseased organs and diseased cells, you're also typically going to be using H&E. I am going to talk about some other staining methods too, that are fairly common in biology, but this is the one you're probably going to run into a lot. So as I said, H&E consists of two different chemicals that are used to stain these light microscope slides. The first one's called hematoxylin. And hematoxylin is a base. It is alkali. It has a pH above 7, which means it's going to be attracted to anything that's acidic, anything that has a pH below 7. It's actually going to form a salt. It's going to bind to any acid present in the cell to form what we would call a salt. And one of the most common acidic structures inside any cell is DNA. If you think about what the abbreviation DNA means, deoxyribonucleic acid. DNA is acidic. So is RNA. RNA is ribonucleic acid, so RNA is also acidic. But matter of fact, DNA and RNA are both called nucleic acids. That's the category of biological molecules that they belong to. The word nucleic acid dates back to the 1800s when scientists first discovered hematoxylin and started staining cells with hematoxylin. They knew that hematoxylin was a base and it was going to be attracted to something acidic. And they noticed that the nuclei of cells always stained dark blue or dark purple when exposed to hematoxylin. So since hematoxylin is a base, they knew that there's something in the nucleus that's acidic. So that's where the word nucleic acid comes from. It just means acidic stuff in the nucleus. And it wasn't until decades later that they found out, oh, there's something in here called DNA. All they knew there was something acidic in the nucleus. So when you stain something with hematoxylin, that's one of the key things that you see when you look at it under the microscope, is that the nuclei of cells stain dark purple or dark blue because they attract a lot of this basic or alkaline hematoxylin stain. Another good example of staining with hematoxylin is there is a type of white blood cell known as a basophil. And when exposed to hematoxylin, there are little granules in the cytoplasm of basophils that stain dark blue because they're attracting hematoxylin. The word basophil literally means loves base. So since hematoxylin is a base, the name basophil actually comes from the fact that this particular class of cells loves base. They're attracting the hematoxylin stain to stain these cytoplasmic granules dark blue. The other part of the H&E process is then using eosin, which is acidic. Now you can't just mix hematoxylin and eosin and stick some bunch of cells in it, because the hematoxylin and eosin, having opposite pH, they're going to neutralize one another. So you have to stain your sample with one of these, flush out any excess stain, then you got to stain it with the second one afterwards. So it's a two-step process to stain with hematoxylin and eosin. So eosin is acidic, so it's going to be attracted to anything that's a base, but it also happens to be attracted to proteins in cells. Eosin has a red color, so any large concentration of proteins is going to stain red. And one of the classic examples of that is the red blood cell, the erythrocyte. An erythrocyte is basically just a big bag of protein. Red blood cells don't even have a nucleus and lose the nucleus. They just are a big bag filled with hemoglobin, which is a protein, of course, that's responsible for carrying oxygen in the blood. So when you stain red blood cells with eosin, they stain bright red because all that eosin is attracted to that giant bag full of protein. And one thing I wondered about, and I should look this up and see if I can find an answer to this, is, are red blood cells named for the fact that we know that they're the cells in blood and blood is red? Or did red blood cells get named for the fact that they stain bright red with eosin? Because eosin was also discovered in the latter part of the 1800s. So I've never known, do we call red blood cells red because blood is red or because they stain red in eosin? Or did somebody decide, oh, wow, there's this stain that stains red blood cells red. Let's use this because it's going to make it really easy to identify red blood cells in a microscope slide. So I don't know what came first. The cytoplasm of, I was going to say most cells, probably all cells, has a lot of protein floating around in it. There are all kinds of proteins that are floating around in the cytoplasm of cells, although they're not usually densely packed the way the proteins are in an erythrocyte, red blood cell. So for most cells, the cytoplasm actually stains pink because it's attracting the red eosin stain, but the proteins aren't that concentrated, so that the red is kind of diffuse. So it gives it kind of a pink color instead of bright red. And then there's an example of another type of white blood cell known as an eosinophil. And these, similar to a basophil, eosinophils have cytoplasmic granules. But the cytoplasmic granules in an eosinophil, the granules are actually basic, they are alkaline. So those granules attract the acidic eosin, and so the cytoplasmic granules in an eosinophil stain bright red. And that's where eosinophils get their name. You know, back when the H&E staining method was first invented, people noticed that you stain blood with H&E, and some of them have blue cytoplasmic granules, they love base, and then others have these bright red cytoplasmic granules, they love the eosin. So hence eosinophil or loves eosin. So here is a typical photomicrograph, typical microscopic image of some cells stained with H&E. And one of the first things you can spot right away is all the little purple dots. Every one of those little purple dots is most likely a nucleus. Then the next thing you notice is that there are a bunch of irregular geometric shapes that are all kind of pink. If you look closely, you realize that there are all kinds of little structures in here. They don't have any kind of uniform, regular geometric shape, but they're all surrounded by little white areas. All these little geometric shapes in here that are pink. That's the cytoplasm of some kind of cell. Now, this is actually skeletal muscle. Some of you may not be able to see it. There's a tiny label at the bottom of the image. This is skeletal muscle, and this skeletal muscle has been cut perpendicular to the direction of the muscle fibers. So all these little geometric shapes that you see down here that are all light pink, those are the ends, the cut ends of the individual muscle cells or myocytes within this skeletal muscle. So you're just looking into the cytoplasm of the cut ends of all these myocytes. And then towards the upper left, we have some other membranes surrounding the muscle. So you have things like some of the fascia and other membranes that cover the surface of the muscle. But down here in the lower part, in the lower right part, we're looking at the cut section through the muscle tissue itself. Just a little bit of trivia. If you look closely at all these little pink geometric shapes, which represent cut sections through the individual myocytes or the individual muscle cells, you might be surprised if you really stop and think about it really hard at the number of little purple dots surrounding each one of those myocytes. Each one of those little geometric shapes represents a cut section through an individual muscle cell. Why are there multiple purple dots surrounding almost every one of those muscle cells? And I don't know, does anybody know the answer to that?
Doesn't skeletal muscle have multiple, like if the cells have multiple nuclei?
Yep. Skeletal myocytes are multinuclear. They have lots of nuclei. So even though we're looking at just one little cross-sectional slice, I mean, we're not even seeing the whole length of these muscle fiber cells. We're actually seeing multiple nuclei associated with each individual muscle fiber or each individual myocyte. Here's another classic example of a tissue stained with H&E. Once again, all those dark blue, dark purple dots that you're seeing are nuclei. You can see a large cluster of bright red cells over here on the left. That represents a slice through a small artery or an arteriole. It's not small enough to be a capillary, Capillaries usually have very small diameter that's usually only just a little bit bigger than the size of a single erythrocyte. So the fact that we've got, like dozens or hundreds of erythrocytes packed into this space means that this is either a small artery or an arteriole. But you can see how all of these erythrocytes, all these red blood cells, stain really bright red because all that hemoglobin is attracting the eosin stain. And then there's a membrane surrounding the artery or arteriole. So this would be the endothelium. These would be thin, flat cells. You can see the pink cytoplasm, and you can see an occasional nucleus within some of these endothelial cells surrounding the artery. Then over on the right-hand side, I don't know if you can see the label way down at the bottom. I know some of you are following along on your own computers. You see that this is labeled lung. The white spaces over here on the right are the alveoli. Now they don't look like big round sacs the way they're usually portrayed in most illustrations. Part of that's because alveoli are not perfect spheres the way they're often illustrated. I mean, I'm guilty of that. I illustrate them that way too, but in reality, the alveoli are not perfect spheres. But the other thing is that when you do the slicing, the thin slicing of these samples, there's usually a lot of distortion, especially something as delicate as an alveolus. It has such a paper thin wall that the alveoli get distorted. So, but these empty spaces are the interior of individual alveoli in the lung. And if you look just to the right of the one big white space where the word lung is located, you'll see there's a thin layer of tissue that's staining kind of light pink, light lavender color, and there's a few nuclei in there. These actually represent the pneumocytes, which are the cells that form the walls of the alveolus. And then all of the red are more red blood cells located within the capillaries that surround the alveoli, because there are networks of capillaries surrounding each individual alveolus. And that's where you get the gas exchange that takes place with the absorption of oxygen into the bloodstream and then carbon dioxide coming out of the bloodstream into the alveolus to then be exhaled. So you can actually see very tiny arterioles and even individual capillaries. Some of them have ruptured open. You got some red blood cells kind of spilling into the alveoli, just again, because of the damage that's caused during the preparation of the slide, but you can still tell that there are little vessels embedded in the walls of the alveoli. Here you see a great example of the three different types of granulocytes. So I mentioned before, there are things called basophils and eosinophils, which are white blood cells. Whoops, why is that not showing? Oh, there it is. So I mentioned, these white blood cells called basophils and eosinophils that attract hematoxylin or attract eosin. These are all a type of white blood cell that's known as a leukocyte, or even more specifically, these are all known as granulocytes, just because these three types of cells have granules in their cytoplasm. And in the case of a basophil, the granules are acidic, so they attract base, so they attract the hematoxylin, and it stains the cytoplasmic granules dark blue. If you look really closely, you might be able to distinguish where the nucleus is. The nucleus in a basophil has kind of a double-lobed, bilobed shape, so you can just barely kind of make out the nucleus, but the nucleus is really obscured by the fact that you've also got all these dark blue staining granules in the basophil. In the middle, we have the eosinophil, which have basic granules, alkaline granules that attract eosinophil, which is acidic. So here it's a little easier to make out the nucleus. It's also a bilobed or multi-lobed nucleus, the two purple areas. And then you can see that there's a lot of red stuff in the cytoplasm, which would be the cytoplasmic granules that are attracting eosin and staining dark red. And then finally, the third group of granulocytes that I didn't mention before are known as neutrophils. And they're called neutrophils because they do have granules in the cytoplasm, but the granules are pH neutral. They have a pH of 7 or roughly 7. So the granules don't attract base and they don't attract acid either. So the granules in a neutrophil don't attract the eosin or the hematoxylin. And so you can still barely tell that there's something in the nucleus. There are granules of some kind, or excuse me, not in the nucleus, in the cytoplasm, but they are pH neutral, so they don't attract eosin or hematoxylin. So that's actually where the word neutrophil comes from. They are neutral loving, or they don't love either acid or base. All right, moving along, another relatively common staining technique in medicine. You don't see it illustrated very often, but I know it is widely used in clinical medicine for a lot of important medical tests. It's the Papanicolau stain, or Pap stain. It's a staining method that's used to detect cells either in what's called a smear, where they take a sample of cells from some tissue in your body and they smear that onto a microscope slide and look at it with the microscope. Or the Pap stain is also used for detecting the presence of cells in bodily fluids, such as looking for the presence of cells in cerebrospinal fluid. Cerebrospinal fluid's not really supposed to have cells in it. But if you have meningitis, you're very likely to have white blood cells in your cerebrospinal fluid. So you take a sample of cerebrospinal fluid from the lumbar spine, through a lumbar puncture, or sometimes called the spinal tap. And then you look at that CSF, you stain it, and then you look at it under a microscope to look for the presence of cells. Sputum, which is just, spit or mucus. Pleural fluid, somebody has an infection in their lung, you might take a sample of pleural fluid and look for the presence of cells in pleural fluid. Synovial fluid from a joint. If people have a lot of white blood cells in the synovial fluid, that's indicative of a form of arthritis in the joint. So you can analyze the presence of cells in these different fluids or in a smear. The staining technique is named after Dr. Papanicolau, and I forget his first name. But he invented the staining technique, plus he's also responsible for what's known as a Pap smear, which anyone who's had a routine gynecological exam is very likely to have had one of these. They take like a little plastic spatula and scrape some cells off the tip of the cervix and spread that on a microscope slide and look at them under the scope, because they're looking for changes in the epithelial cells of the cervix as an early detection method for cervical cancer. And that's also named after Dr. Papanicolau. It's used to detect the presence of cells themselves, but also to distinguish different kinds of epithelial cells from one another. The Pap stain uses H&E, hematoxylin and eosin. So the hematoxylin still stains the nuclei dark blue or dark purple. The eosin will stain red blood cells red, but any kind of superficial epithelium, so epithelial cells that are close to the surface of an organ, will stain pink. The cytoplasm will stain pink, excuse me. But then there are two other stains that are also used in the Pap smear or the Pap stain. This is what distinguishes it from just regular old H&E. There's a light green or a fast green stain that stains deeper epithelial cells. So cells that are coming from deeper within an epithelial membrane, they'll tend to stain green using the light green or fast green stain. And then there's another orange stain, this G6 orange stain, that stains keratin, which is a protein, of course, that's found particularly in the epidermis. You have cells in your epidermis called keratinocytes that produce lots of keratin. keratin is also the protein that makes up your fingernails and your hair. But also G6 stains glycogen. So glycogen is a long chain of carbohydrates that your body uses to store sugar. So here is one example of Pap stain. And let's see. Well, first of all, you can see that all of these cells have nuclei, and the nuclei are staining dark blue or purple because of the hematoxylin, the H&E part of the staining process. We've got some pink cells that are staining pink because of the eosin, and these would be superficial epithelial cells. You got some green ones where that fast green is staining. These would be deeper layers of epithelial cells. And then I think there's one orange one in here towards the upper left. I'd call that orange. And so that's attracting that orange G6 stain. So that's probably something like a keratinocyte that has some keratin in it. So that's actually a mixture of different cell types that you can distinguish in this one sample. Even though the cells look pretty nondescript, you know, they don't have obvious shape. You can distinguish the different cell types based on their color because of the staining method. Another staining technique that you're very likely to see in biology and, you know, in the medical field, especially if you are looking at images about nerve cells, is the silver stain. And there are a couple of different variations on the silver staining method. It was originally developed by, gosh, I forget his name, Golgi, Camillo Golgi, back in the 1800s, invented the silver staining technique and allowed him to stain neurons. The neurons will stain either a kind of a reddish brown or even black, depending on the particular variation of the stain. So Camillo Golgi was one of the first people to recognize and describe and illustrate nerve cells using the staining technique. You also probably know Golgi's name because the Golgi apparatus or Golgi body in the cell is named after him. There's another variation on the silver stain that's often referred to as the Cajal stain. It's named after Santiago Ramón y Cajal, who was a Spanish neuroscientist working in late 1800s, early 1900s. Really very, very famous neuroscientist. He was one of the first to really describe the structure of different kinds of nerve cells in the central nervous system, especially. So he would stain, you know, cross-sectional slices through different parts of the brain and very carefully describe and characterize the branching pattern of the different neurons in all different parts of the brain. And it turns out he was a pretty clever artist too. That's one of his illustrations on the right. That's an illustration of what's called a Purkinje cell, which is, I believe, the most common cell type found in the cortex of the cerebellum. And come around over here to the side. Several years ago, there was an exhibit of I'm screwing up his name. Here in America, we can't wrap our heads around the fact that the Spanish countries, it's very common for people to have multiple last names. So his full name is Santiago Ramón y Cajal. The E is the letter Y, which is Spanish for him. So his name is actually Santiago Ramón and Cajal. So he's really got kind of two last names. But like I said, in America, we can't wrap our heads around that. So but anyway, a number of years ago, the AMI held a meeting outside of Boston, and it just so happened that year MIT was hosting a pivotal article showing, you know, all these particulars of the nerve cells. And it was really cool that I went to that and saw it. So those dispersions. But they're also cool. his drawings of the nerve cells. And it's cool to look at, just, aesthetic. It's kind of interesting, but then, the science behind it is pretty cool. So this just reproduces lots and lots of these drawings. So an important person in the history of scientific administration, as well as being an important person in the history of neuroscience. So I'll have that. You're going to go to interest in this. Okay. All right, so that's all silver staining method, for looking at, in particular, looking at neurons, especially in the central nervous system. Here is another technique that's also used primarily for looking at the nervous system, actually exclusively for the nervous system, because this is a technique that stains myelin. So it's a stain called Luxol Fast Blue, and it stains myelin that's present in the central nervous system. I guess it could probably stain myelin in the peripheral nervous system too, because a lot of the axons in some of your peripheral nerves are also myelinated too. So it's just always going to stain myelin blue. It's usually combined with H&E. So for example, this picture here is showing a cut section through the pons, and it's stained with a combination of hematoxylin and eosin plus Luxol Fast Blue. So if you look at some of the cell structures in here, you see the characteristic, dark purple nuclei, you see some pink cytoplasm, but then all that blue stuff is all myelin. In the upper right, you can actually see like elongated strands of myelin. So you would actually have axons that were traveling, sort of from upper left to bottom right. The diagonal strands you're seeing are myelinated axons. But then on the far left and towards the bottom of the illustration, you'll see some blue, but they look like little black dots. Those are also axons that are myelinated, but we're looking at a cut section, cut perpendicular to the axis of those myelinated axons. That's why they look like little blue dots, because we're looking at the cut ends of those myelinated neurons. All right. So all those stains that I mentioned are ways of enhancing light microscopy so that you can actually see something when you look at samples of cells under a microscope. There's another group of techniques that is quite a bit newer, probably within the past 20 to 30 years is when this has come into existence. Many of those stains I mentioned date back to the 1800s, but fluorescence is something that's much newer. So this is another way of really enhancing light microscopy, making it easier to see things that you otherwise couldn't see under a regular light microscope. So the way fluorescence works is you have some substance, some molecule, something, and you shine a light on it. You bombard that thing, whatever it is you're trying to look at, with a particular wavelength of light. It doesn't even have to be visible light. It could actually be something like ultraviolet light, but you're hitting a sample with some wavelength of electromagnetic radiation. And the object that you're shining the light on is going to absorb some of that energy. It's going to absorb energy from that light, but then the sample is going to start to glow. It's going to start to emit energy, but it emits the energy at a longer wavelength, at a different wavelength. So you're shining one color of light on the sample then glows in a different color. And a great example of that is you could shine ultraviolet radiation onto a sample, which is not visible to the eye, so you can't see the UV that you're shining onto the sample, but then the sample glows green in response. It's glowing at a lower wavelength. And then you can detect the presence of that green glow. and it'll tell you, something interesting about the thing that you're looking at. So there are three broad categories or three types of imaging that take advantage of this concept of fluorescence. The first of these is what's called autofluorescence. And what this is referring to is the fact that there are some molecules located in your cells that will fluoresce on their own without having to do anything special to them. Now, you still have to shine light on them at a certain wavelength, and they'll glow a different wavelength, but you don't have to treat the substances with anything special. You don't have to put any kind of stain or dye in there. They'll just fluoresce all in their own. You just shine a light on them and they glow. So there are some proteins, such as collagen and elastin. These are proteins that are very common in cartilage, but also found in the extracellular matrix of a lot of tissues. These things will just glow spontaneously in the presence of light. Some of the molecules that are involved in energy transport in the body, such as NADPH and FAD, or FADH. NAD is nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. Both of these are involved in, like, Krebs cycle, bringing electrons to the electron transport chain for the generation of ATP. So they're involved in the energy production mechanism within the cell. There's also chlorophyll, which obviously you wouldn't find that in humans, but this is another molecule that will fluoresce all on its own. Obviously, that's found in plants. So as I said, you have these different molecules that are present in the cells in your tissues that will fluoresce all by themselves. But then there's another type of fluorescence. This is one that's really become extremely important in the last 20 years or so, is what's called immunofluorescence. So with immunofluorescence, the idea is to attach a fluorescent molecule to some target structure in a cell or in a tissue or in some kind of sample, some biological sample. So what you do is you have a set of fluorescent dyes, I put that in quotes, they're more accurately referred to as fluorophores. You attach one of these fluorescent fluorophores to an antibody, and the antibody is going to bind to specific molecules in the cell. So let's say you wanted to locate the actin in a cell. Actin is a protein that's an important part of muscle cells. It's part of the cytoskeleton of most cells. And let's say you wanted to figure out where the actin is located in different cells. You could create an antibody that's going to bind specifically to actin, then attach one of these fluorophores or fluorescent dyes to the antibody. You introduce the antibody into a sample, and the antibodies will then attach only to the actin, only to whatever they're antibodies for. And then you shine, like ultraviolet light on the sample, and it's going to glow green. And then you can detect, exactly where the actin is located within these cells. So as I said, you're just attaching a fluorophore to an antibody. The antibody is then attached to specific molecules in the cell. You then image that, and you can tell exactly where these different molecules then are located in the cells. The first fluorophore that was discovered and used for this process of immunofluorescence was something called green fluorescent protein, or GFP, discovered back in the 1990s, so 30 years ago. And it's become extraordinarily important ever since then. GFP was originally isolated from a species of jellyfish. It's one of these glowing jellyfish, I think it's found down in Australia. And turns out it's a very tiny little protein that's really easy to manufacture, and then you can add it to an antibody and then target certain molecules in the cell. There are now other fluorophores that are very common. So there's your GFP up at the top, but there are yellow ones and orange ones and red ones and blue ones and turquoise ones. And so what you could do is you could attach, say, green fluorescent protein to one set of antibodies that's going to target a particular type of molecule in the cell, and attach the cerulean fluorophore to a different antibody that's going to attach to a different group of molecules in the cell, and so on. And so you could actually have numerous different colors of fluorophores that are targeting or marking different molecules within a cell. So here's an example of that. These are light microscope images with the addition of fluorophores that have been attracted to particular molecules through this process of immunofluorescence. So on the left, you have some vascular endothelial cells. So these would be the flat cells that line the inside of your arteries and veins. And they obviously attached a blue fluorophore to an antibody that was specifically targeting something in the nucleus. More than likely, it's targeting like the histone proteins in the chromatin in the nucleus. So there's this bright blue glow in the nucleus because these fluorophores have bound to something in the nucleus. All these green fibrous strands, that's all actin. So they probably attached green fluorescent protein to an antibody for actin. And so all those antibodies are stuck to the actin in the cell. Actin is an important part of the cytoskeleton. So it's forming kind of the skeleton or framework of this cell, giving the cell its shape. Those actin cytoskeleton filaments also help cells move if a cell moves around. And then the red, this is probably that cherry red fluorophore that's attaching to something on the cell membrane, probably some surface protein on the outside of the cell. And so you get this bright red glow around the outside of the cell membrane of all these different cell types. On the right, you see this is, I don't know exactly what tissue this is, but I do know that each of these ovals is a small artery located within the tissue. And the green, once again, is targeting actin in the smooth muscle cells in the wall of the artery. So muscle cells have a lot of actin in them because that's one of the contractile proteins that's present in all muscle. And so you're seeing the green fluorophore that's been attached to the actin proteins in the walls of these smooth muscle cells in the wall of an artery. And then there's obviously some other fluorescent fluorophores in here as well. There's some blue ones and some red ones. Yeah. Oh, okay. No. I love this picture here. I mean, what an amazing view of a cell undergoing cytokinesis, a cell in the process of cell division. So it's basically finished with mitosis and it's starting to split apart. So there's some green fluorescent protein that's attaching to something in the cell membrane to stain or make the cell membrane glow bright green. There's a blue attached to the chromosomes. So you've got the chromosomes that are being segregated into one cell and chromosomes being segregated into the other cell. So something, like the cerulean or turquoise fluorophore on the chromosomes. And then the bright sort of gold color, that would be a fluorophore that's attached to an antibody to tubulin. It's the protein that makes up the microtubules. So you're able to actually see the remnants of the mitotic spindle where the chromosomes were pulled apart. So as you can see, you create some really, really dramatic images that would be really useful if you were trying to illustrate this process of cell division. I think this one's just really pretty. I put this on my wall at home. I just think it's a cool image, but it's an incredibly powerful image, from a scientific point of view, because it's staining so many different structures. There's a whole bunch of different fluorophores used in here to detect different proteins that are expressed in different tissues within the wall of the stomach. So this is the gastric mucosa. And if you look up at the top, you see the bright red fluorophores attracted to the columnar epithelial cells that are lining the gastric pits. These are the gastric glands. But then there are other glandular cells that extend deep down into the mucosa. And you see there's even differences in the expression of different proteins. The deeper you go into the gastric mucosa, you're getting a lot more of these bright pinks and stuff, indicating, the expression of different proteins at different levels. But, it gives you a great view of the cellular structure of the gastric mucosa. One final use of fluorescence. I'll talk about this and then I'll let you all take a break because I'm talking a long time. But so we've talked about autofluorescence. So certain molecules in the cell that just fluoresce all on their own without having to add anything special to them. We've talked about immunofluorescence. So, using antibodies to attach fluorophores to specific molecules in the cell. This is another really interesting use of fluorescence, what's known as a reporter of gene expression. The way this works is, let's say you want to figure out which cells in the body are expressing some particular gene. So we've got this target gene here represented by just this little chunk of DNA double helix. And let's say you want to know which cells in the body actually express that particular gene. So what you do is you take the gene for a fluorophore, the gene for one of these fluorescent proteins like GFP, and you insert that gene into the regulatory region of the gene that you're interested in, and you create a new hybrid gene. So you've actually got the gene for the protein that you're interested in with the GFP gene spliced into it. That gene is then going to be transcribed into messenger RNA and translated into protein, and you will manufacture a protein which has the normal protein that you're interested in, but it actually has the green fluorescent protein embedded in it as part of the protein. And you then get a microscope slide and you look at that microscope slide, you bombard the sample, probably with ultraviolet light, and it's going to glow green in response. And it's going to allow you to see which cells in some part of the body are actually expressing this gene that you're interested in. So here's an example on the right showing a slice through the cerebral cortex of a mouse. And they've inserted this GFP gene into a gene called MAPT, which codes for a protein called tau. And the reason they were doing this, I'm sure, is because they were probably studying Alzheimer's disease, because tau is a really important protein in understanding the pathology of Alzheimer's. You get what are called tau tangles, which are clusters of tau protein that are found in the cells that are damaged in Alzheimer's disease. So understanding exactly where this tau protein is expressed, or where the tau gene is expressed in the brain, is really important. So you can actually tell from this image that there's a lot of tau protein expressed in the cells that are very close to the surface of the cerebral cortex, in the very outer part of the gray matter. But then as you go deeper into the cerebral cortex, you have cells that are expressing less and less of it, and you go deep enough and you get no expression of this MAPT gene, no production of tau. So, I'm sure that's an important clue in helping us to finally understand the pathology of Alzheimer's disease. So, okay, so why don't you all take a short break, maybe come back at say 3:35 and we'll start promptly then to talk about something else. Why is it showing that? That's annoying. Okay, there we go. All right, so moving on to another imaging modality, another imaging technology that helps us visualize cellular structures and really small things is electron microscopy. So a very different form of microscopy. I mentioned before that light microscopy has been with us for hundreds of years. Electron microscopy is much newer, less than 100 years old. And when it was first introduced in the 1930s, it was not very good, but it's improved in quality dramatically over several decades. Electron microscopy takes advantage of the fact that electrons have a much smaller wavelength, a much shorter wavelength than visible light. So remember I said that visible light has a wavelength around 10 to the minus 7 meters, between 400 and 700 nanometers, which means that visible light can only see things down to about that size, down to about 10 to the minus 7 meters. Electrons have a much shorter wavelength. Their wavelength is down around 10 to the minus 9 meters, down around the size of a nanometer. So electron microscopy allows us to see things not quite down to 10 to the minus 9, but down to about 10 to the minus 8 meters. So electron microscopy allows us to see things that are about 1/10 the size of what you can see with a light microscope. So this extends the visibility of structures down to the level of organelles, bacteria, and viruses. So things that are smaller than individual cells. I'm not going to go through the technology of exactly how electron microscopy works, but I will mention that it does have some difficulties or some shortcomings. First of all, it's really hard to do. Electron microscopy takes advantage of the fact that structures are conductive to electrons, they're conductive to electricity. But you don't want things to be so conductive that they absorb all of the electricity. So for one thing, in order to do electron microscopy, you have to get rid of all the water. So any kind of structure that contains water, which includes all of our cells, you've got to get rid of the water, but you have to get rid of the water, you have to dehydrate the sample, but you have to do it in such a way that retains the shape and structure of the object that you're trying to image. So let's say you've got a bunch of cells, and if you simply dehydrate them, if you just dry them out, the cells are just going to shrivel and crumble into a powder. You won't be able to see anything. So you have to get rid of the water, but do it in such a way that you don't lose the shape of the structures. So they do this by replacing the water with some other solvent that is not conductive of electricity. So I'm trying to remember exactly what some of them are off the top of my head. I think maybe ethanol, ethyl alcohol. There may be some other, acetone, I think, is another one. So you have to somehow find a way to replace the water in a sample with something like acetone that won't conduct electricity, so that the cells save their shape, but without actually containing water. Then you want to coat the surface of the structure with something very thin that actually will conduct electricity. And usually they use gold. You use a little machine called a sputter coater that takes a tiny little sample of gold, maybe 25 cents worth of gold, which is not very much. And it turns that tiny sample of gold into a vapor of individual gold atoms. And those individual gold atoms then settle onto the surface of whatever it is you're trying to image, and creates this super thin electrical conductive coating on the surface of the sample that you're trying to image. Right above our heads, practically almost literally right above our heads, the College of Art and Design used to actually have an electron microscopy lab. I think the lab is still there. The problem is they had a couple of really ancient electron microscopes that, after they were probably 20 or 30 years beyond when they should have just been retired. They finally just all broke down, and they couldn't afford to repair them or replace them. So Ted Kinsman, who's in the photo school, he's a faculty member in the photo school. He actually used to teach a couple courses in electron microscopy, and he used to give our class, this class, we used to take a little tour up there, and he would show us how he would sputter coat something in this little sputter coating. He'd put like a nickel or something in there and sputter coat the nickel with a thin layer of gold atoms. But unfortunately, we don't have that resource anymore. Another downside to electron microscopy is that all of the images are grayscale. You're imaging things with electrons, you're not imaging things with visible light, so there is no color. So everything appears grayscale. You can see highlights and shadows, you can see three-dimensional form, but there's no color in an electron microscope image. So there are three broad categories of electron microscopy that are used for looking at different things. First of all, there's what's called scanning electron microscopy, or SEM. This allows you to scan the external surface of a three-dimensional object or a group of three-dimensional objects. So this is where that dehydration and sputter coating is especially important, because you have to maintain the shape of the structures and then coat their surfaces with that thin layer of gold atoms. SEM does not have the best resolution, though. You can't see things that are super tiny. The resolution of SEM usually goes down to about 10 to the minus 7 meters, which is the same as light microscopy. So SEM won't let you see things that are any smaller than you could already see with a light microscope. But it does have the advantage of being able to see things as an actual three-dimensional image, which is actually really cool. It's a great reference for illustrations if you're trying to illustrate the 3D structure of a cell or a tissue or something like that. The next form of electron microscopy is transmission electron microscopy. And this is similar to light microscopes in the sense that you have to cut a thin slice through a tissue, but then it allows you to image that slice at a higher resolution. So this will go down to about 10 to the minus 8 meters. So about 1/10 the scale of what you can see with a light microscope. And then finally, the most recent development in electron microscopy that's probably come about within the past 20 years is what's called cryo-EM or cryo-ET, which stands for cryo-electron microscopy or cryo-electron tomography. This is very similar to regular electron microscopy, except for the fact that it's performed at extremely low temperatures. And you don't have to sputter coat these things with gold. When you cool something down to extremely low temperatures, it becomes conductive. I mean, anything becomes conductive when you cool it down to a low enough temperature. In fact, many things are what are called superconductors when they get down to extremely, extremely cold temperatures. They can conduct electricity very easily. So to do cryo-electron microscopy and cryo-electron tomography, you just have to cool the samples down to ridiculously cold temperatures. And if you're familiar with the Kelvin scale, 0 Kelvin is what we call absolute zero. It's the temperature at which all atomic motion stops. So cryo-EM and cryo-ET are performed at around 100 Kelvin, which is like negative 300 Fahrenheit, something like that. Very, very cold. But this allows you to see even smaller things down to about 10 to the minus 9 meters. So down to the scale of a nanometer. So basically down to molecular scale. So here's an example of a typical SEM, a typical scanning electron micrograph. So this is the view of the inside of a bronchus. And it's not super magnified. I probably should have put the scale on here, but I don't remember what the scale was for this. But you could magnify this more with SEM. You could even see something smaller with a light microscope. So this is not very magnified, but you get this beautiful three-dimensional landscape. So you can see the microvilli of the ciliated cells lining the inside of the airway, the inside of the bronchus. I'm not sure what all the round lumpy cells are. I believe the the kind of star-shaped ones, the ones that are kind of radiating, I think those are macrophages. But so we're seeing a bunch of different cell types on the surface of all these ciliated endothelial cells, or excuse me, epithelial cells lining the inside of a bronchus. Here's a really high resolution scanning electron micrograph of the surface of an E. coli bacterium. So that big thing in the background is an E. coli. And these lighter colored things on the surface of the E. coli are bacteriophages. Those are viruses that are infecting the E. coli bacterial cell. So you can see down to virus scale with electron microscopy. Here's a fun little image by Ted Kinsman, who I was talking about before, who used to run the electron microscopy lab here at RIT. He still teaches here, but he teaches other areas of scientific photography. And this is a scanning electron micrograph of the bottom of a marijuana leaf. Ted has photographed and written an entire book about marijuana under the microscope. And so what you're looking at here is, like I say, it's the bottom of a cannabis leaf. And the little stalks with the little purple balls on the end are what are called trichomes. And the little purple balls are actually little blobs, little droplets of THC, tetrahydrocannabinol, which is the active ingredient, which is why some people enjoy, this particular plant. Now, you might remember earlier I said that electron micrographs are always black and white. This is clearly not black and white. This is all colored by hand. So this originally was a grayscale image. And he actually, for a while, he used to hire our students to colorize his images for him. So you just have to select the little blobs at the end and color them purple, and color the rest of the leaf green, and then there's some other things colored red. But so all the color in here is all completely artificial. And just in case anybody's looking for a Christmas present, here's the cover of Ted's book, Marijuana Under the Microscope. I don't get kickbacks from that. Here's an example of a transmission electron micrograph. So this is where the sample is sliced thin, and you're looking through it, very similar to the way you'd look through a thin slice with a light microscope. This is an individual cell. This is what's called a plasma cell that is significantly enlarged, so much so that you can really start to see some of the details of the individual organelles. So the large round thing, not surprisingly, that's the nucleus. Plasma cells are the cells, they're actually derived from B cells, and they are basically little antibody factories. The whole role of a plasma cell is to just start cranking out huge amounts of antibody, which is a type of protein. What you're seeing in the nucleus, the black clumpy stuff, that's the chromatin. You get this clumping of chromatin that creates, they call it a wagon wheel nucleus. Somebody decided that the white parts look like spokes of an old-fashioned wagon wheel. I don't quite see that myself, but that's very characteristic of plasma cells. And then all of the cytoplasm is just jammed with rough endoplasmic reticulum. All those little squiggles you see out in the cytoplasm are rough endoplasmic reticulum. So just ER lined with ribosomes. So this thing is just cranking out messenger RNA and cranking out proteins all day long to manufacture these antibodies. And then you can see there's an occasional mitochondrion in the cytoplasm, the little ovals labeled M. Right, I'm gonna jump ahead to another type of imaging technology that we're actually going to come back to and talk about in a lot more detail later. I just want to introduce it briefly today. And like I say, we'll come back and discuss it in more detail when we get into the molecular part of the course. But I just wanted to briefly introduce another imaging technology called X-ray diffraction, which allows you to see things that are really, really, really small, like individual molecules, individual atoms, even with X-ray diffraction. It's interesting that X-ray diffraction that allows you to see things at the atomic scale has actually been around longer than electron microscopy. X-ray diffraction dates back to the 19-teens. In fact, it was 1911, I believe it was Max von Laue was the first one to propose that you could image atomic structures with X-rays. And then it was a year later, 1912, that William Lawrence Bragg in London created the first X-ray diffraction image of a crystal of sodium chloride, of table salt. But X-ray diffraction takes advantage of the fact that X-rays have even smaller wavelength, even smaller than electrons. So electrons have a wavelength down around 10 to the minus 9 meters. X-rays have a wavelength down in the range of 10 to the minus 10 or even 10 to the minus 11 meters, which means that X-rays will bounce off the surface of individual atoms. So you can use X-rays to actually image individual atoms or individual molecules down to a scale around 1 angstrom or 10 to the minus 10 meters. So the thing you're looking at on the left is what's called an electron density map, which is basically a map showing the density or showing the location of clusters of electrons. And from that electron density map, you can actually draw the structure of a particular molecule. So dating back even to the 19-teens, we had this technology that allowed us to see really, really little things, down to angstrom scale, 10 to the minus 9, 10 to the minus 10 meters. But light microscopy was limited to seeing things down to 10 to the minus 7. Electron microscopy eventually extended that down to about 10 to the minus 8 or thereabouts. But for much of the 20th century, we were dealing with this unusual problem. And I don't know why the monitor is not showing. There we go. I keep changing slides on my computer and it's not showing up on the projector, which is really weird. But okay, hopefully it's working now. So for much of the 20th century, scientists were faced with this unusual problem called the resolution gap. That meant that we could see things that were kind of big and we could see things that were really, really small, but we couldn't see things in between. So light microscopy that had been around for centuries allowed us to see things down to about 10 to the minus 7 meters. So you could see cells, you could kind of see organelles, but not very clearly. At the other extreme, you had this new technology called X-ray diffraction, introduced in the teens, that allowed you to see things that were really, really small, like 10 to the minus 9, 10 to the minus 10 meters. So the structure of individual molecules, or even the shape of an individual atom. But for much of the 20th century, we couldn't see things in between. We couldn't see things in that gap from 10 to the minus 7 to 10 to the minus 9, which meant, in particular, we had a hard time seeing how the individual molecules were packaged together to create things like organelles and then to create whole cells. So here's a representation of this resolution gap. So if you look up at the top, you see we've got scale, meters, centimeters, millimeters, micrometers or microns, nanometers, angstroms, which would be 10 to the minus 10 meters. You see across the bottom, things that would be measured in this scale. So, a child be about 1 meter, thickness of a human hair about 1/10 of a millimeter, red blood cells around a micron or thereabouts, bacteria, viruses, individual molecules like the size of a DNA double helix, down here around 10 to the minus 9 thereabouts, and then individual atoms down around angstrom scale, down around 10 to the minus 10. So the human eye lets you see things down to about 10 to the minus 4, which is about the thickness of a human hair. Light microscopy overlaps with that somewhat, but it extends our ability to see things down to about 10 to the minus 7 meters, so roughly the size of a bacterium. for much of the 20th century, there was this resolution gap where X-ray diffraction allowed us to see things down around nanometer and angstrom scale, but we couldn't see anything in that gap between them. Electron microscopy, which first started in the 30s and got better in the 40s, 50s, and 60s, began to extend our ability to see things down into that resolution gap. And then finally, just in the past 20 years, with the advent of cryo-EM and cryo-ET, we finally filled in the rest of that resolution gap. So we can now see how individual molecules are packaged together to form things like a virus or an organelle inside a cell. So this is all because of the advent of these new technologies, cryo-electron microscopy and cryo-electron tomography. So I mentioned this before, I'll just repeat it, that the cryo just means cold. So cryo-EM and cryo-ET are just electron microscopy performed at very, very low temperatures, less than 100 Kelvin. So approaching, absolute zero. The only difference between cryo-EM and cryo-ET is that cryo-ET is done in 3 dimensions. So cryo-EM would just give you an individual slice. Cryo-ET actually lets you look at things from multiple angles and reconstruct things as a 3-dimensional object. So cryo-ET in particular has been very useful for medical illustrators to allow us to be able to see how individual molecules actually are packaged together to form structures within the cell. This thing on the left is a cryo-ET of a thing called a perforin, which is a complex of proteins. It's a very large group of proteins that can actually poke a hole in the surface of a cell. And if you look up at the top, you see the original cryo-electron micrograph images that were used to reconstruct this as a 3D model. So what you're seeing on the left is a view of a cell, and that thing up at the top of the cell is actually a side view of this perforin complex that's poking a hole in the side of that cell. And then just to the right of that, we're looking kind of like almost at a top view or an end view of the perforins, and there's actually 3 of them here that are all poking holes into this cell. And so you can actually use those side views and top views and combine them together to create an overall 3-dimensional view of this perforin. The tan is actually a chunk of the cell membrane of the cell, and then the blue represents the perforin protein complex that's created a hole in the cell membrane. Over on the right is another great example of the use of cryo-ET. So this is an image of the complex of 3 proteins, actin, myosin, and tropomyosin, which are the contractile proteins in a muscle that allow a muscle cell to contract. So the little red blobs in the center of the image are individual molecules of actin, and the individual molecules of actin are joined together to form a long chain. In fact, there's 2 chains of actin that are kind of braided together or twisted around one another. It's hard to see that in this image, but take my word for it that there's a double chain of actin molecules that are twisted around one another. The blue stripe is a molecule called tropomyosin that runs the length of the actin fiber, and it's also wrapping around or sort of twisting around the outside of the actin, kind of like the stripes on the outside of a candy cane, coiling around the actin molecules. And then all the other colored things, the yellow, the sort of turquoise blue, the kind of magenta, purpley color, those represent different parts of the myosin molecule, which these are the flexible heads, the myosin heads, the things that actually contract or that actually flex, to create a muscle contraction. So what we're seeing here is the myosin heads actually bonded to the actin molecules And then these myosin heads would then flex to create the contraction of the muscle cell. So we can actually see how individual molecules are packaged together to form that contractile mechanism inside a muscle cell. Another one of my favorite cryo-ET structures, I've actually used this as a reference for an illustration of the ribosome and how DNA translate, or excuse me, how the process of translation takes place at the ribosome. So the ribosome consists of two parts, two subunits. There's a small subunit shown in the pale yellow, and then there's a large subunit shown in the pale blue. Located within the small subunit are 3 binding sites for the binding of transfer RNA, the tRNA molecules. And you can actually see them in here. They're shown in different colors. The orange, or kind of orangey brown molecule back there is a tRNA that's in, I believe that's in the E site, which is the exit site. It's the last position where the tRNA sits before it's ejected from the ribosome. The green one is in one of the other sites. I forget if that's the P or the E. There's an E, a P, and an A, and I forget the order of them. I just know the E is the exit one. So there's a green tRNA, and then there's a purple tRNA in the third site. The turquoise structure up here is actually the messenger RNA. So that's what has the codons on it that are being read by the anticodons of the tRNAs. So you've got the messenger RNA entering the ribosome up here at the top, and then the messenger RNA exiting the ribosome over here on the left. And then there's a little brownish structure down here that's just barely visible. That's actually the protein itself that's being manufactured by the amino acids that are held together by these tRNA molecules. And that's going to pass down through this thing called the translocating PCC, or protein conducting channel. It's going to pass through that PCC into the interior of the endoplasmic reticulum. Here's another one. This is the structure of a microtubule, which makes up part of the cytoskeleton of the cell. It's also the thing that forms the mitotic spindle during mitosis. And it's made up of individual subunits of a protein called tubulin that form just a big coiling stack to create an individual microtubule. So just to summarize what I've been saying here, we've got light microscopy that allows us to see things down to about 10 to the minus 7 meters, allowing us to see, whole tissues and individual cells. And you can enhance your light microscopy with different forms of staining or with different fluorescence techniques. Electron microscope extends our resolution down a little bit smaller, down about 10 to the minus 8 meters. That allows you to see organelles more clearly, bacteria and viruses. Cryo-EM and cryo-ET take that down another order of magnitude, down to about 10 to the minus 9 meters, to show how molecules are actually packaged together to form things like organelles or viruses. And then finally, X-ray crystallography allows us to see the smallest things, down to about, I should say, 10 to the minus 10 meters, down to things at the atomic and molecular scale. And we'll come back and spend a lot more time talking about that later in the course. So I'm going to shift gears now that I've talked to you about some of these different technologies, and talk a little bit about actually illustrating cells and a few tools that you can use to help you create accurate illustrations of cells. So I've given you some references here that you can look up to help you with illustrations of cellular structures. And let's see if I can jump over to that. And it's not showing up there, so I may have to stop share and try that again. It's still not showing up correctly on screen. Okay, that's not what I want. Where is? Maybe if I get out of full screen in PowerPoint, maybe it'll do what I want it to do. It's just, it's weird. I'm seeing one thing on my screen, something else on the projector, and hopefully the people on Zoom are seeing the right stuff too. Let's try this. So I have some of these resources loaded up that I wanted to share with you. The first of these is called BioNumbers. And it's just, you know, the URL is up here. It's in the PDF of the PowerPoint that's posted up on my courses. It's basically a database of numerical information about cells. So if you wanted to know the volume of a red blood cell, you could probably look that up. If you wanted to know the diameter of a red blood cell, you could look that up. Let's just see if we put in a search term like erythrocyte. Hopefully I spelled it right. And see, okay, there's a volume of an erythrocyte. There's the lifespan. How long do they live? Here's how much hemoglobin is found in an erythrocyte. And of course, you know, they have it for different species too. So if you wanted the volume of a typical erythrocyte, you'd really want to look at this one specifically for humans. I don't know if it lets you sort. No, it doesn't let you sort them by organism or not, but so you get some interesting numerical information about cells, which may be helpful when you're illustrating, especially if you want to know something like what's the relative scale of an E. coli bacteria to like a typical red blood cell? You know, what are their relative sizes? I'm sure you could look that up in here. They also have a book that you can get that has all these numbers in it. And there are links to, you know, how the book is laid out. So you can just read all this information about, you know, all kinds of information, everything you'd ever want to know about viruses and about their relative size and scale. I put an older version of this book. You can actually download the whole thing as a PDF, but I think the only one you can download is an older 2015 version. But I put a copy of that in my courses for you. You know, I'd encourage you to download it and take a look at it because it's a great resource. Okay, but that's all I wanted to show you about this BioNumbers site. Another website that's pretty useful for getting references for cellular structures is this Cell Image Library. And this is just, you know, tens of thousands of images of cells. And there are all different kinds of images. There are light microscope images, fluorescent microscope images, electron microscope images. There's even some animations in here where they animate or videos, you know, showing like cell division taking place, you know, in a video. You can search the Cell Image Library by cell process. So if you're interested in some cellular process like cell division, or I don't know if you could look up, I don't know if you can look up something like electron transport or some physiological process, but like if we click on this, you know, cell adhesion, cell death, like, you know, process of apoptosis, cell migration and motility, here's cell division. If you click on any one of these, you see just all kinds of images of cell division. You know, there's 83 pages of different images of cell division. You can look it up by cell component. So, you know, there's 655 images of just the actin cytoskeleton, 32 images of cell walls, 853 pictures of chromosomes. So if you're looking for good reference for, you know, what do neurons look like, you've got 687 different images, electron micrographs and all kinds of other images. There's some fluorescence images of neurons. You can look up by cell type. You can narrow it down by organism if you want to look at, just humans, or you want to look at, Drosophila, the fruit fly, or something like that. So anyway, I'm not going to dwell on this. I just wanted to kind of quickly introduce you to this resource. Another one it's kind of cool. I haven't found it super useful, but there's this thing called the Allen Cell Explorer from the Allen Institute for Cell Science out in Washington State. And you can search their whole cell collection. You can also look at their digital tools. They have something called a 3D cell viewer, or they have this thing called the Deep Cell Zoom. Let me look at the 3D cell viewer, because it's kind of an interesting little tool. Okay, go away. So it's just a library of three-dimensional images of cells that have all been imaged using fluorescent imaging. So I could actually pick this little cell up and move it around. And, it doesn't look like much, but I think one thing you realize quickly when you go through this 3D cell viewer is that cells are not necessarily the simple shapes that we think of them as. Like this one has a rather irregular shape to it. But you can clearly see the nucleus. You can see something is fluorescently labeled green. And actually, if you look over on the right-hand side, you can see what these things are. So this is called Sec61 beta. The pink is the cell membrane. The blue is the DNA. You can hide the different fluorescent, images. And so only look at one thing at a time if you want, or you can just look at the membrane. It'd be kind of nice if you could actually download these as 3D models and then see if you could somehow use those in a 3D program. But you can download them as TIFF images, which are just static two-dimensional images. But I haven't figured out a way to actually get the data in 3D. So it's an interesting thing to explore cell structure, but haven't found it all that useful, in a practical sense for creating illustrations. Last one I want to show you is this EM data resource, or EM data bank. This is a database of electron microscopy data, and you can just do a simple keyword search. So let's say we wanted to look for, I know, I'm going to look for ribosome. I'm going to see if I can find that model of the 3D structure of a ribosome. So we get 2,327 different 3D models of ribosomes. I have no idea which one that one is. I mean, there's page after page of these things. So here we're just seeing 20 entries. We could, view 50 or 100 entries, or we could view all 2,300 of them. But if I were to click, say, on this one, I can see a preview image of what the thing might look like. I can see the blue is probably the small subunit, the yellow is probably the large subunit, and then the green, purple, and darker blue are the tRNA molecules. So I get a sense for what's in the model. But let's see if this will work or not. There's a 3D viewer to view the model. So I can actually view it in 3D in here. But I know that there are ways of changing. I click on a little tool icon, and I can hide different aspects of the model. Okay, that just hit everything, so that's not good. Right. So you can explore different aspects of it. What you're actually seeing like the molecular structure superimposed. All these little geometric shapes you're seeing are bits of protein and RNA, because the ribosome is actually made up of a combination of RNA and protein. I think it's like 60 or 70% RNA and 30 or 40% protein, all packaged together. So you're seeing all of that stuff here. The electron data bank used to be a little bit more powerful, a little bit more useful for us, because you could actually download these in 3D, and then we had a plugin that allowed you to then open the 3D model in Cinema 4D and actually like model it and render it in Cinema 4D. But this is the plugin that doesn't work anymore, unfortunately, and I haven't found a good replacement for it yet. So so be aware that there is this 3D electron microscopy data resource out there, and at some point, hopefully, I'll find a new tool. I mean, there is a thing under here for tools, but maybe I'll explore this EM software list sometime to take a look at it and try to see if I can identify some different tools for looking at electron microscopy data. All right, well, let me jump back to the PowerPoint. Hopefully this will share properly on Zoom and on screen. All right, that's good. All right, so I just went through these resources, and the these resources will help you illustrate molecular structures. You may be able to use some of these to get a sense for the overall three-dimensional structure of a cell. But another important question when illustrating cellular structures is what color to make. And that might not seem like that important of a question, but when you stop and think about it, it's actually not necessarily a simple question to answer. Because remember that when you look at images of cells under a light microscope, usually they are completely clear and you can hardly see anything unless you stain them. So the only way you can see structures under a light microscope is to stain the sample or use fluorescence or something like that. But you're not seeing the actual color of the cells or the tissues that those cells come from. You're only seeing the color of the stain. So keep that in mind, that the colors that you often see, in microscopic images of cells are not the actual colors of the cells themselves. Remember that electron microscope images are always grayscale. They're always black and white. That picture I showed of the cannabis leaf from Ted Kinsman was all bright and colored, but that was all artificial. He added that color just to make the image more visually interesting. But you don't get a clue for what the color of an actual cell is from looking at an electron micrograph. So you have to decide, how do you want to color the cells? And there are some popular approaches to choosing the colors for cells when you do cellular illustrations. And I want to show you some examples of these different approaches to how you decide what color to make your cells. One of the most common approaches to coloring cells is to color the cells the same color as the organ or the tissue that the cells come from. So if you can see what something like a liver looks like, in a cadaver or in a surgical photo or something, if you see what real liver color looks like, if you want to illustrate liver cells, make the liver cells the same color as the liver. And so that's what you're seeing here. You're seeing hepatocytes, which are the liver cells, and they're all colored kind of reddish brown. which is a color that we typically associate with the liver, especially in a living person. So you're seeing a lot of detail here about the structure of the liver. And just as a little teaching moment here, you might remember from gross anatomy, we talked a lot about the portal venous system. And we talked about something called portal hypertension. There was an essay question, one of the exams about portal hypertension. So all of the blood coming from all of the digestive organs in the abdomen enters a vein called the hepatic portal vein. That hepatic portal vein goes into the liver. And this is a branch of that hepatic portal vein illustrated right here. Well, the blood from that hepatic portal vein then goes off into all these little branches inside the liver, little spaces called sinusoids, which are little spaces between the hepatocytes, between the liver cells. You can actually see that magnified over on the right. That dark area is a sinusoid with a little vein traveling through the sinusoid between the hepatocytes. So all of that venous blood is actually filtered by the hepatocytes. So any toxic chemicals that are present in the blood are neutralized by enzymes in the hepatocytes. The hepatocytes absorb glucose from the blood and convert that glucose into glycogen to store it for later. The hepatocytes also absorb lipids and cholesterol from the blood, coming from your digested meal. And then the hepatocytes regulate the levels of different lipids or lipoproteins in the blood. So you're actually seeing how the blood actually has to get filtered at the cellular level. Then eventually those little veins coming out of the sinusoids are gathered up by larger veins that then flow back into what are called the hepatic veins that then dump back into the inferior vena cava. All right. So that's just repeating a little anatomy lesson. But the point of this is just to show that liver cells are colored to look like livers. This is a terrible illustration, but it's a good example of the concept. These are supposed to be muscle cells. They don't look a whole lot like muscle cells. Maybe I'll give the artist a little bit of a credit. Maybe these are supposed to be smooth muscle cells, which have a little bit more of a spindle shape like this. But this is a good example of what I call gratuitous 3D. I did it in 3D because I know how, not because I should. But I put this in here just to show that we think of muscles as being red, so the artist illustrated muscle cells being red. Here's one from my own work that I know I've shown this to the grads a couple times. The undergrads may not have seen this before, but this is an illustration of the pathophysiology of celiac disease, like gluten sensitivity in the intestine. And the key thing here is that, I'm illustrating the epithelial cells that line the inside of the digestive tract. And so I colored all of those intestinal cells in a kind of a peach color to look like intestine. Here's another example from the liver. We don't normally think of the liver as being that kind of gray color, but if you look over on the right, you'll see that this artist illustrated the liver as being a kind of a gray color. I think the artist actually made a very smart choice here. The artist colored the liver gray so that it didn't compete for attention with the gallbladder and the bile ducts, which are really the focus of the illustration. So the artist used a vivid color for the gallbladder and bile ducts, so that attracts your attention. The liver is left this kind of muted, dull gray color. But over on the left side, the hepatocytes, the liver cells, use that same liver color. You notice that you've got what are called bile canaliculi, the little canals where bile is produced, and then they flow into larger and larger bile ducts, and that's all colored green to match the color of the ducts and the gallbladder in the right-hand image. So again, we're matching the color of the tissue that the cells come from. So that may seem, that's a pretty simple idea, but it's something you have to think about. You have to decide, do I want to color the cells to match the tissue that they're coming from? Another very common approach is to color cells according to the way they stain. If since H&E is the most common staining technique, it's very common to see cells illustrated using H&E staining methods. Now, these are 3D models. These are not real cells, but the artist in this case chose to render the cells with colors that are reflective of the staining technique. So you've got, in every case, you've got a blue or a purple nucleus because of the hematoxylin. In the case of the basophil, that's the one with the dark blue or dark purple granules because it attracts hematoxylin also. You've got an eosinophil over on the left with dark red stained granules because they attract eosin. You've got the neutrophil, which would be nice if you could actually see there are granules in the neutrophil, but they're hard to see because they're not staining because it's pH neutral. You've got a monocyte down here on the left, which doesn't have any granules. We've got a lymphocyte, like a B cell or a T cell, and then you've got erythrocytes staining dark red because they attract a lot of eosin also. Here's my version of exactly the same thing. This is, a pretty simple illustration, but done in Adobe Illustrator, showing exactly the same stuff. So an erythrocyte in dark red, eosinophil with bright red granules, basophil with dark blue granules, neutrophil with kind of pale neutral granules, and then lymphocyte or B cell and T cell, and then a monocyte. So this idea of matching the staining is another popular method. And one thing I should have put in here is usually when I illustrate neurons, I'll always illustrate them yellow. Now that's partly because we think of nerves as being yellow, so it makes sense to color neurons yellow, but oftentimes I'll put a lot of brown stuff in the cell body of the neuron. There's something called Nissl substance, which are these little granules in the cell body of a neuron. And I'll often make them kind of brown, which sort of matches the silver stain, the Cajal stain that's used to stain neurons kind of reddish brown color. Another approach that's very common, I don't necessarily highly recommend it, but okay, there we go. Boy, this is really lag time with the projector for some reason. Is what I've nicknamed the water balloon approach to illustrating cells. So a lot of people imagine a cell as being a thin membrane filled with water. The cytoplasm of a cell is probably about 70% water. So we imagine the cell as being just this big, empty water balloon, this big bag full of water. And then, of course, there's a nucleus floating around inside the cell somewhere. It's probably not the most realistic approach because even though the cytoplasm is about 70% water, that other 30% really kind of fills up a lot of the space inside the cell. The cell is actually very full and very crowded with all kinds of molecules and things. So the water balloon rendering kind of perpetuates this myth that cells are basically just big empty bags of water. But a lot of artists use it. I found this pretty easily. Just do a Google search for cells, and you're bound to find some water balloon representations of cells. And I'm guilty of it myself. There was a long time actually where when I did any kind of cellular illustrations, I would always just render them blue because we think of water as being blue. We associate the color blue with water because you look at the ocean, it looks blue. Sometimes even ice will have kind of a bluish tint to it because of the way it refracts light or absorbs light. So I'm actually going to walk you through a little history of the way I've illustrated cells over the years. There's a whole series of pathology textbooks that I've been illustrating for over 25 years. The flagship of the series is called Robbins' Pathologic Basis of Disease. It's been around for decades. There's a smaller book, companion volume called Robbins' Basic Pathology. There's a newer one that just came out a year ago called Robbins' Essential Pathology. And then there's some others. There's even a veterinary pathology book. And I've been illustrating all these books for over 25 years. If you go back to some of the first editions that I illustrated, the first 5 editions were illustrated by somebody else. And I think they probably had 4 or 5 different illustrators, and the artwork was kind of a hodgepodge. So then when they were getting ready to do the 6th edition, the publisher came to me and said, we want you to redo all of the artwork for the whole book so that it all has a consistent style and a consistent look. So I redid all the artwork for the 6th edition of the book, and then, created new artwork for the 7th edition. This is going back into the 1990s. And I chose this kind of water balloon style for no particular good reason other than I thought of cells as being little water balloons, little bags of water. Now this one shows some aspects of the immune system. It's comparing innate immunity and adaptive immunity. And you can actually distinguish the different cell types if you look carefully at their nuclei. So there's a monocyte or a macrophage over here on the left with this kind of kidney bean nucleus. There's a neutrophil here that has the trilobe nucleus. There's a natural killer cell down here. And then we've got lymphocytes, we got B lymphocytes and T lymphocytes. And, to someone who's actually studying pathology, they would actually recognize the differences in these cells just based on the size and shape of the nucleus. Well, I forget what year it was, but I went to a meeting in Chicago with the authors and representatives from Elsevier, from the publisher. And we spent 2 whole days in Chicago just talking about the book and planning the next edition, the 8th edition of the book. And I threw this slide in here just to be kind of funny, but it's kind of, I don't know, it's kind of serious too. If you've ever wondered why textbooks are so expensive, this is why. So you got the 3 authors over there on the left, Dr. Kumar, Dr. Abbas, and Dr. Astor. You got 4 representatives from the publishing company, and then of course yours truly. And we are standing in the kitchen of Charlie Trotter's restaurant, which was the fanciest restaurant in Chicago at that time. And when you go to Charlie Trotter's, I think it's closed since then, but when you used to go there, they only had one menu. It's not like you ordered off a menu. You got their grand menu. So you got little tiny tastes of all of those things. Tempura of Nantucket Bay scallop, Santa Barbara sea urchin with Buddha's hand. I don't even know what that is. Arkansas rabbit loin and all this stuff. And if that wasn't bad enough, the publisher said, hey, let's get the wines that accompany each course. So for each course, they would bring over like 2 bottles of wine and you'd get a different wine with every course. So the bill for dinner was like $2,500 for 8 of us. So that's why your textbooks are so expensive. But it was a lot of fun. But anyway, the point of this is that we spent like an entire Saturday locked in a room just talking about this book. And at one point, Dr. Kumar, who's on the bottom left there, he's the lead author. He says to me, Jim, one question, why blue? And he was asking about the cells. And I told him the whole water balloon thing. And he's like, to be honest, of all of the colors I would have chosen, I would not have picked blue. It's like, okay. So back to the drawing board. We got rid of the whole water balloon thing and went with, let's color the cells according to the staining method. Since H&E is by far the most common stain used in pathology, let's match the staining technique. So we have dark purple nuclei and pink cytoplasm for the different kinds of cells. And so this approach lasted for 2 more editions, the 8th and 9th edition. So now we're up into the, well up into the 2000s. And had another meeting. They didn't take us to a fancy restaurant this time though, but had another meeting with the authors. And they said, we're getting some feedback from the students that the students are having trouble telling the different cell types apart, even though the nuclei are very distinctive and very recognizable. Let's try something else. Let's color code the different cell types. So that's yet another approach. Same illustration for the 10th edition. And it's not showing up eventually. There it is. Let's come up with a different color scheme for each cell type to make it easier for the students to tell them apart, which makes a lot of sense, honestly. So the only one that kept the H&E color is the neutrophil, but phagocytes and dendritic cells got this kind of yellow-orange thing, NK cells, brown, B cells, blue, T cells, pink, and so on. So that was the last one, and I haven't heard about the 11th edition yet. I'm still waiting to hear about that one, so we'll find out if they change their mind again. One more example of the same thing, going back to the original editions that I illustrated. Water balloons, 8th and 9th editions, matching the stain, H&E staining. Now, with the 10th edition, they also made another request. I guess one of their competing pathology books started using some 3D art. And if you've heard me talk in class, you know how I feel about 3D. I got no problem with 3D. I just think it's overused because people think it's cooler, even if it's not a good choice. So they were kind of pushing me to create some 3D images for the book. And I resisted the idea of going full-blown 3D, but we came to a compromise. Let's at least take the artwork into Photoshop and give it a little bit more depth and a little bit more drama. So, that's the same basic illustration, same concept, just now give it a little bit more depth by taking the Illustrator file and bringing it into Photoshop and just painting on it a little bit more. So we did like one illustration for each chapter in this newer sort of 3D style. And last thing I will leave you with are just a couple of other kind of unusual approaches, but really interesting approaches to coloring cells. Way back on the very first day, I brought in an example of this guy's book. This is David Goodsell, who is a molecular biologist at the Scripps Research Institute in Southern California, but he's also a really talented painter, talented artist. And he creates these really dramatic cellular landscapes where we're looking at the interior of a cell magnified, like a couple million times. And so we get to see the individual molecules that make up the cell. And I forget what this one is over on the left. I know there's a little yellow rope in here, which would be DNA. But I forget exactly what we're looking at here. I do know this one, we're looking at a close-up of a ribosome. So we actually see the DNA in the ribosome because ribosomes have their own genetic material. Not ribosome, mitochondria, sorry. We're looking at a close-up of a mitochondrion because mitochondria have their own genetic material, their own DNA. So you're actually seeing the individual proteins that are embedded in the inner and outer membranes of the mitochondria. And I actually recognize one. There's one that looks like a little mushroom with a little bar next to it that I'm circling. That's ATP synthase. It's the molecule that actually makes ATP in the mitochondrion. And David Goodsell was also part of a developing a little piece of software, which if anybody wants to search for it, you can just do a Google search for Cell Paint. It's a fun little program that actually lets you paint with molecules. So you get little brushes that let you choose like a cell membrane brush or a protein brush, or here's some actin filaments inside of a cell. There's an actin filament brush, and you can just kind of paint, make a little virus for yourself or make a little cell. All of the kind of orange and brown things are proteins that would be found in blood plasma. So there's different kinds of antibodies in there. There's some IgG and some IgM antibodies, different immunoglobulins floating around in there, as well as just like plasma proteins floating around in the blood. So we've got, I think that's an HIV virus floating in blood plasma about to infect a T cell. So one of the little pink receptors on the T cell is CD4, which is the receptor that the HIV uses to get into the T cell. So anyway, so we're probably should have mentioned this at the beginning, but we will have our second quiz in the class on Monday, which will be all about these different kinds of microscopy and staining techniques and fluorescence and all that kind of fun stuff. So that's it, and actually finished 5 minutes early for a change. So I'll see you all on Monday.